Hi class, we are on chapter 26.6. This is the last section. This is a very short section, but it's just talking about the applications of coordination compounds, which is a very big deal. Coordination compounds impact every part of life as we know it in different things. I've used some of these coordination compounds in my lab when I was in a researcher, specifically chelating agents. So applications of coordination compounds, extraction of metals from ores, because a lot of the metals are not found just as metals themselves. They are in ores and they're not easy to pull out from those ores. They have to go through a process. So using coordination compounds to pull out, for instance, silver and gold is one of the ways that metals are pulled out of the ore. Another is pulling nickel out of a nickel complex. Chelating agents, remember chelating agents are very stable compounds, and EDTA is, which I've used when I was a student in one of the labs, and it is used as a for a poisoning. A person ingests lead. Lead is a very big deal. It's very dangerous. And with an EDTA, which is attached to a calcium complex, the calcium and the lead will switch places, thereby attaching the lead to the EDTA. And calcium, of course, is something that we need in our body, so that's not a big deal. But the body will then excrete the lead complex, and it's now non-toxic because it has left your body. And calcium is something we need, so it works. So EDTA is a good example of a ligand that can attach to metals, so a coordination compound. Chemical analysis, we can determine we have a thiocyanate groups, and depending on which metal it's attached to, we can get a blue solution or a red solution. Another is also for chemical analysis, a nickel palladium, which is attaching to a dimethylglyoxamine, and I have a picture of that, the bottom one, coming up. So transition metals, you can see here is a list of biomolecules and how they work. We have chromium, that works with insulin to control the utilization of glucose. Manganese, I always mess this one up, molybdenum. Yes, it is. It's molybdenum. It's involved in hemoglobin synthesis. Iron, obviously with iron oxygen transport. Copper, involved in hemoglobin synthesis and so forth. So we do have traces of transition metals and so forth, and as long as they're not, overabundant in our body, they are important to keep us functioning. Here's an example of hemoglobin. Hemoglobin transports oxygen via a coordination complex, and they're usually very big, like this. You see this is hemi, and it involves 4 nitrogens, 1, 2, 3, 4, and it attaches to the iron 2 complex. And the N atom from the hemoglobin protein is the 5th ligand, leaving the 6th ligand position in the oxygen. I'm not going to really go too much into more of that, but you can see a visual of how this big compound, this coordination compound, holds on to these metal ions in them. And if in the absence of oxygen, hemoglobin absorbs a yellowish green light near 560 nanometers, giving red blood cells a dark red color. And if oxygen-containing hemoglobin absorbs light near 540 nanometers and 570 nanometers, it has a bright red color than oxygen-free hemoglobin. And I really didn't focus on the colors in this section too much, more about what actually was going on with these compounds. Okay, so if you go back here to this last one, if a certain ligand has a preference for certain metals, and if it does attach to these metals by coordinates, it will give off a different color based on the precipitate will be a different color. So then you'll know if you have nickel or you have palladium. So DMG is, excuse me, dimethyl oxamate. Excuse me, I said it earlier, dimethylglyoxamate. DMG, we'll just call it. And you can add this ligand into solutions and it will prefer, so selective ligands prefer specific ions. So depending on the precipitate, you can determine if nickel is a part of the solution or palladium is a part of the solution. And what precipitates out will tell you if you have red will be a nickel complex and with attaching to coordinating with the DMG, or palladium attaching to the DMG, and it will give a yellowish precipitate. Coloring agents are also iron blue is a mixture of hexacyanoiron 2 and iron 3, used in inks, blueprinting, cosmetics, and so forth. We can also use for biomolecules. A porphyrin ring, and porphyrin rings, there's different There are different porphyrin rings, so they have different substituent groups connected around the outside. This one you can see, this porphyrin is a square planar. We have the nitrogen, so you can see 4 nitrogens are attached, and it can coordinate and have that metal in the center right here. But you see it's kind of big and bulky in this, but so it coordinates to a central atom, a central metal ion. We have chlorophyll. Chlorophyll is attached here, you can see in the middle to this porphyrin ring. And remember, chlorophyll is responsible for the absorption of light to provide energy for photosynthesis. And there's a magnesium ion held in the middle of the porphyrin ring, so for photosynthesis. And then we have carbonic anhydrase, and this is used to, if I remember right, it's used it involves respiration. So you can see we have a zinc in the middle of this, tetrahedral complex zinc. And then at the end, this one's actually very interesting because has to do with an anti-cancer drug. And I know I'm probably not saying it right, cisplatin or cisplatin. And there are 2 different types of this. So remember, if we go back to geometric isomers, there's a trans and a cis. But what they found is that the cis actually works and the trans does not. And the trans, they think, works because it's straight from your book. It's believed to function by attaching itself to the cancer's DNA and replacing the CA ligands with donor atoms from DNA strands. So that's the cis arrangement of the CA ligands, but that doesn't happen with the trans. So even these geometric isomers are very important for us to understand how to use them. At my job, when I worked in the lab, we were working with diagnostic and therapeutic drugs. That's what we were working on. And how do we know which drugs work? Well, we actually deal with a lot of these fancy looking complexes that get really, really, they are complex a lot. And we need to know how things fit into it so it can attach itself to where we're looking for in the body and soul. So a lot of this stuff it's really important. And if you go into certain types of chemistry or even other research, you will definitely cover a lot of this more.